28-year-old Peter Dervan arrived at the California Institute of Technology with little evidence he would soon create a new field of science.
It was 1973. I had no research proposals and only 6 months of postdoctoral experience at Stanford. In fact, the breadth of my early publication record demonstrates that I was casting about for a new research direction.
Very little was known about where and how compounds bind to DNA when Dervan began his seminal research.
I became interested in creating novel molecular shapes with different properties than those found in nature.
Though they didn't yet exist, he understood such molecules could have enormous potential to control whether and how certain genes are expressed within cells. The affinity cleaving process he eventually developed identifies DNA sequences bound by drugs and other compounds, and has led to many of today's natural product anti-cancer agents. A scientific founder of Gilead Sciences, he has also helped launch a novel class of commercialized antivirals, even as he looks forward to the next stage of his own work.
Looking ahead, I visualize an era of transcription therapy, where this basic research could underpin a new class of human medicines.
Throughout his many career achievements, Dervan has remained a committed teacher and mentor.
During those first years at Caltech, I realized that by teaching my subject matter, I should be moving in bolder, more audacious directions, and so I picked the interface of chemistry and biology. Now, as I look to the future, I see extraordinary intellectual vitality and excitement. Several of my former coworkers have followed my path into academics, and it's remarkable how each one has created his or her own scientific vision.
A designer whose palette is the very stuff of life, Peter Dervan continues to create at the interface of chemistry, biology, and human medicine.